Okay, block 1 and 4 biology. Hopefully you guys gave your best effort on this. I am at a track meet in Lincoln, so I got this recorded. This is getting you ready for your osmosis and diffusion quiz, which is tomorrow, which is Friday. I do think some of the lettering was off on some of these, so make sure as I go through the answers here, I expect phones away. Yes, all of you. Even you, Abel, in fourth block, put it away. If I messed up, make sure the lettering, make sure you have the right answers down and understand why. So, going over our bell ringer and the gummy bears and everything, which term is used to describe the carrying, italicized words are always important, of molecules across a membrane that does not require energy. So I know it's not active transport because that requires energy. This one says it does not require energy. Osmosis is water. This just says molecules. So between diffusion and facilitated diffusion left, if we're carrying it across, that means it needs help, which would be the facilitated diffusion. So number one should be facilitated diffusion. Number two is a concept we learned yesterday. Both of these are some of those proteins that help things across the membrane. The main difference being that channel proteins have just one little tunnel going straight down the way, whereas carrier proteins are ones that will usually change their shape in order to let things in or across. They're both proteins that help things across, but the carrier protein changes shape, whereas the channel protein just is one nice tube across. Number three is a very tough one, but like I told you guys the other day, you need to learn to draw pictures. So if you go draw a little picture over here, you would want a protozoan's a little amoeba-like thing. If it's pushing water out of itself, that would mean that it's saltier outside the solution than inside the solution because water's going out. If it's saltier, more concentrated outside, number three should be hypertonic, and you should have a drawing that supports that because it's going to where the Gatorade is strongest. Number four, osmosis we know has to do with water. So if I go look, the only one that talks about water is letter D, and it says it will equal the concentration by moving water particles. So number four should be letter D. Number five, you'd need to draw a picture. I draw a little box, and then on one side I put A, 40%, and on the other side I put B, 20%. It says molecules are allowed to cross from side to side, and I know diffusion says things will move from high to low. So it will move from the 40% to the 20% until they equal out, which means things will move from A to B, which is our first option here. Some helium will move from chamber A to chamber B because we'll go from high to low. On the back side, more practice. A hypotonic solution, remember hypoglycemic means we're low on blood sugar, so hypo is less concentrated than the inside of the cell, which usually means that the inside is greater and water will go in. Number seven, turgor pressure we learned yesterday. Turgor pressure is just the natural internal pressure inside of a cell that may come from water or other things, and it's very important in plant cells and how they are taller and less wilty during the day. It's internal cell pressure, just like a balloon. Number eight, diffusion takes place. If we go look, it does take place through a bilipid, but that's probably not the only way because we know if we spray the wonderful perfume from Meredith or Desi that it doesn't have to just be a lipid bilayer. Same thing with your gummy bears. It is not from low to high. It is not just liquids. However, diffusion does always go from high to low. So number eight should be from high to low. Number nine, on the other hand, asks me which is not a characteristic of facilitated diffusion. This is where I need help. So, does it require a carrier protein? Yes, it does. Does it move against a gradient? No. It moves with the gradient because we're still going from high to low. It requires no energy input. That is usually true. And letter D says it moves through the lipid bilayer directly. That is where it's wrong. It doesn't just move directly through there. It needs help from those proteins. So that's kind of the weird word at the end there that makes letter D not true. Last but not least, draw yourself a little picture, but if there's more oxygen in blood, it will move out of the blood, right? It'll go from high to low. So it will go from the blood to the organ because it will go from high to low. So With that being said, that takes care of those bell ringers. First block, pass yours up to a Darius or whoever's there. Second block or fourth block, pass yours up to who sits in the corner? I don't know who sits up in the corner, whoever that is. Pass them up and give it to the sub. Next, you guys will watch a short video about endo and exocytosis. So at this point, please get your notes out to page 6. You will watch a short video and take notes about that. Then on the board, to the left of me, I guess, where you're looking, is an example of the diagram worksheet I told you we'd practice, where you have a word, the definition, and a diagram of it, and working those things together. You are each going to make your own, but you can work, actually you can work on that with a partner. Everything else today will be by yourself. So the sub has the answer key at this point. He can take the purple sheet down so you guys can't see it. Do not glue or tape until you're done. The glue and scissors should be out on the island, as well as the tape. And then you guys are just going to go piece by piece through what I left on the board. Make sure we're practicing and getting ready for our osmosis quiz tomorrow. Thanks for being rock stars. I will see you guys Friday. Use your class time accordingly.